mRNA vaccine platform was first developed in the early 1990s to target influenza and HIV, among other diseases. All of that research paved the way for the efficacious COVID-19 vaccine. Now, with mRNA thrust into the limelight, its potential has a lot of people talking about where it may ultimately take medicine. Moderna recently launched a phase 1 clinical trial for HIV. The other big focus now is cancer.
Scientists are exploring if the new mRNA vaccine technology that brought us safe and effective COVID-19 vaccines could also be used to stop cancer cells when they first arrive.
Perhaps it makes sense then that the minds behind the Pfizer-BioNTech mRNA vaccine actually consider themselves first and foremost cancer doctors. I recently sat down for a virtual chat with BioNTech co-founders Özlem Türeci and Uhar Sahin from their headquarters in Germany. Now, before we get into its future, let's take a look at its foundation. What exactly is an mRNA vaccine and how does it work? Messenger ribonucleic acid, mRNA, is genetic material found in our bodies that instruct our cells to produce certain proteins. In the case of COVID-19, the mRNA vaccine prompts our cells to make the coronavirus spike protein, which then triggers our immune system to recognize a critical piece of the virus and create antibodies to fight it off in the future. mRNA technology to treat diseases like cancer is a bit different. Those vaccines have to be tailor-made for each specific patient's cancer cell markers. But the goal is the same: help our bodies create an immune response to better do its job.
You know, when we hear the term vaccine, you think of it as to prevent disease, right? It's to prevent disease. When we're talking about it in cancer, we're talking about treating an existing tumor. You're talking about personalized cancer therapy here. If there's a particular target that you choose, how do you know it is specific really to that cancer and not to healthy human cells in that particular patient's body?
The way to find this out is to compare with normal tissue. For mutations, we do this by comparing to the patient's blood cells, which represent the normal, not mutated counterpart and are representative for normal tissues.
And what is exciting is that in a clinical trial, the immune response which is generated is really recognizing the tumor cell but not the normal cell.
BioNTech currently has human clinical trials for two of the hardest cancers to treat: colorectal and pancreatic cancer. Could mRNA be a key to treating these diseases?